Next up, we have Curtis Huttenhower from Harvard University, who's going to talk to us about multi-omics for the human microbiome and its interaction with the built environment.
Excellent, thank you. So I'll try to juggle both hands here and a timer, and I think we have the AV working this time. Everyone here is at least thinking about microbial communities in the context of the human body or the built environment or both. So I won't have a lot of introduction, but I want to touch on 3 stories today that I think are relevant to the considerations at the meeting here. First, I'll speak a little bit to the Human Microbiome Project, the second phase of which just wrapped up as a really sort of deep focused study of human-associated microbial communities. I'll talk a little bit about methods for characterizing novel microbial community sequence diversity. I know a lot of people here think about the built environment, say, oh, it's easy to study human bugs. We know everything about human-associated microbes already. That's of course not entirely true, and we're getting pretty good at understanding mechanisms of novelty in microbial communities, the bugs that are less well studied. And then if there's time at the end, I'll mention just very briefly some of the work we're doing to look at interactions between the human microbiome and the built environment, especially as it comes to viable microbial transmission. So again, I won't have a lot of introduction since I want to show a little bit from all of these studies today. But to speak to the Human Microbiome Project first, the second phase of the HMP, which I'll refer to as the HMP2, just wrapped up recently and differed quite a bit from the first phase that was published several years ago, which has been sort of the HMP and the literature and the language of the field for a while. The HMP2, rather than studying the microbiome in health, like the HMP1 focused on, had a mandate to look at microbiome dynamics as they interact with the human immune response and other human cellular and molecular responses over time in disease. And so that gave us an environment in which to really drill deeply into human-associated microbial communities, their dynamic function using multi-omic, many different measurement techniques and many views of the microbiome. So we did this in the context of inflammatory bowel disease. I won't really speak to IBD specifically today, but it's a gastrointestinal inflammatory disorder that comes in 2 main clinical subtypes. So in order to study the IBD gut microbiome using a variety of different multi-omic methods, we followed just over 130 individuals, about half Crohn's disease and a quarter ulcerative colitis, again, those 2 main clinical subtypes, and a quarter non-IBD controls for 1 year each. So this was a way to get dynamic information over time on the microbiome in the presence or absence of an immune perturbation, essentially. And these 130 individuals were spread not just across these inflammatory phenotypes, but over 5 different clinical centers and a range of ages and of disease carriage time. So some of these were recent onset and some of them were established disease patients. So we really got a wide swath of different phenotypes, inflammatory phenotypes, to study along with the microbiome and host responses. And so to see the microbiome and host responses using as many different views and lenses as possible, our main sample type was stool collected every 2 weeks for a year. So we did, relevant to the spaceflight microbiome, we worked to develop a home sampling kit. It was as convenient as possible. These were very dedicated patients that we're very appreciative to, while also enabling a pretty wide range of different microbiome and host measurements. So we studied mainly stool samples every 2 weeks from these individuals, coupled in this case with colon biopsies taken at enrollment at baseline from a couple of standardized sites along the colon, and blood draws taken roughly quarterly, every few months. And so these together gave us the sort of NIH mandated many different views of the host microbial system. We were able to derive mostly microbially targeted data from the stool samples, including sequencing and otherwise, metagenomics, metatranscriptomics, viral sequencing, and more functional profiling like metabolomics and proteomics. And then from the biopsy and blood draw samples, we got mostly host data in red here, including also some sequencing, exome sequencing for genetics, bisulfite sequencing for epigenetics, RNA sequencing, especially in the biopsy samples, and then again, some other functional measures like serology for circulating antibody levels. And so the overall study design let us get synchronized time points for most individuals and most sample types and hit those samples with as many different measurements as possible. So we had the hoped for multi-omic views of the host and microbiome dynamically over time during perturbations from the disease. One thing I think is relevant in this context is that all of the resources from the HMP2, both our project and the others under the HMP umbrella, are all publicly available. So in our case, this includes both information on the overall data coordinating center and our own web portal, the IBD MDB, IBD Multi'omics Database. So that's not just data, but things like protocols, sample collection, exactly how we built those kits, questionnaires, the clinical instruments that we use to get dietary data, medication data, how we handled the samples, how we made data. All of those are finally now in a publication as of a few weeks ago and available online, as are the raw and processed data. So any of these multi-omic data types, from raw sequencing through processed microbial or host gene or transcriptional profiles, are available here along with all of our metadata. The IRB allowed us to release that, with the exception of human sequences that have to go to dbGaP. So they're still publicly available through controlled access. So this, along with the HMP1 healthy data, provide a really good baseline resource for comparing new populations to either sequencing in the HMP1 or now sort of a wide range of microbiome and host omics in the HMP2. So again, I won't go a lot into the IBD specific results today, but this lets us do sort of what's more standard microbiome epidemiology now, testing individual feature types in host or microbial responses to see how they associate with inflammation. And that includes not just bugs, but our more functional measures like identifying classes of compounds that are enriched or depleted in the gut during inflammation. So something like the entire set of sphingolipids are jointly significantly enriched during both Crohn's disease and ulcerative colitis. The entire set of lactones, for example, are significantly depleted in both of the IBD subsets. In terms of providing new platforms with which to analyze the microbiome and microbial communities, I think things get more interesting, though, in combining these different data types, either comparing individual measurements or starting to look at them at a more systems level, like we'll see later on. So one example here, in starting to stitch these different measurements together, compares microbes that were individually associated, either enriched or depleted, in the gut microbiome during inflammation, with small molecule metabolomic chemical measurements that were, again, enriched or depleted in the gut during inflammation. And this heat map shows correlations between the two after regressing out some of the covariates that we didn't want. So we didn't want these comparisons to be affected by age or medications or antibiotics, for example, but also in this case, actually regressing out as much as we can the effects of the disease itself. So these are bugs and small molecules that are co-varying both across individuals and over time, even more than you'd expect just from the effects of inflammation. And that starts to suggest potential mechanisms by which a bug and a small molecule might want to co-vary in an environment, either because the bug is generating that small molecule, or it depends on it metabolically, or it could be interacting with another microbe that generates or depends on that small molecule. And you can see the strength of some of these associations in this one example between Ruminococcus navis and docosapentanoic acid. The levels of the organism and the compound are highly correlated in the gut, independently of disease, but they're also both enriched in these red and yellow points, which are Crohn's disease and ulcerative colitis, again, respectively. And so you can start to tease apart these potential hypotheses about mechanisms by drilling into some of the sequencing data, in this case, metagenomically. And again, to skip some of the background, we're able to identify, for Ruminococcus navis in particular, a subset of strains that were uniquely disease-associated, that were uniquely present in individuals with IBD. What this plot shows is each column represents one microbial community, one gut microbiome. We've called out just the individuals, either controls or IBD patients, who were carrying Ruminococcus navis at all. And then on the rows, we show a subset of about 200 genes that uniquely defined the group of Ruminococcus navis strains that were unique to the IBD patients. And so you can see these genes tend to occur in blocks that are roughly operonic. You'll often get strains that gain or lose a chunk of genes at the same time. If you squint real hard, you can see what some of the labels on these gene rows are. The subset of genes where we have some functional characterization often point to things like handling of redox stress, which is one of the defining characteristics of the inflamed gut, iron acquisition, also a limited resource in the gut environment, adhesion or invasion, so these are bugs that want to press closer up to the epithelium. But most of the rows here are not labeled. I think this is really interesting in as much as we know now that this is a set of strains that are uniquely associated with a health outcome and corresponding small molecule metabolism. We know which genes define those strains, and in the vast majority of cases, we don't really know why, functionally or biochemically. And that's a general property of microbial communities, be it in the gut or otherwise. If we look even out, in this case, at healthy examples from the HMP1, the first human microbiome project population, on average, about a quarter of the gene families that we see have some reasonably good biochemistry. They do have a confident function assigned to them. About a quarter tend to be really novel. So they're so diverged that they're not really like anything that we have in an existing gene catalog. And the remaining 50% or so are somewhere in between. They have a sequence that is assignable to a known gene family, but not necessarily with any confident biochemistry. This is in the gut, which, to be fair, is a very well-studied environment where we know a lot about which microbes are there and what they're doing. And things, as many people I'm sure here who study the built environment are familiar with, things get harder and harder the further away you go from well-characterized environments. So this is challenging in as much as it means it can be hard to explain why, now that we can see phenotype-specific, strain-specific, gene-specific biochemistry in microbial communities, it can be hard to explain exactly what that is in terms of gene function. So that's the downside. The upside is that it means that we all have a lot of work to do for a while still. So this is job security for a little while. To transition into some of the examples of how we're working to better characterize this type of microbial dark matter, Nicholas Agata's group just finished up a project that we worked with them on, looking at pulling as much novel sequence as possible out of microbial communities and then organizing that in a way that you can better understand it later on and use it to better contextualize additional new microbial communities. So in this study, we put together a combination of just under 50 different shotgun metagenomic studies. They're all human associated, but not just the gut. They're mostly gut microbiomes, but from overall across the body. This totaled just under 10,000 individual samples. And we ran them through an assembly pipeline, so to try to keep everything, whether or not it has a good reference sequence or not. We ran them through single sample assembly, followed by binning, so just kind of very vanilla metagenomic assembly pipeline at this point. And then imposed a quality threshold on the resulting metagenomic assembly bins that's similar to that used for high quality isolate genomes. So across these thousands of metagenomes, this resulted in about 150,000 reasonably good assemblies and about 80,000 bins that were arguably as good as many isolate genomes. In comparing, 70,000. In comparing these 70,000 with the 80,000 or so isolate reference genomes that were available when we started this study, we put these 150,000 microbial genomes together, clustered them down to about a 5% divergence level, and called that roughly a species. This gave us around 5,000 species-level genome bins, or SGBs, that were human-associated. About 1,100 of these contained a known reference genome. Surprisingly, the remaining 3,800 did not. So they're species-level clades for which we can get a pretty good, at least one, and often thousands of pretty good assemblies from somewhere in the human microbiome. And then another 11,000 or so bins contained non-human associated isolates. These are species that we never see associated with the human microbiome. And so this kind of surprising novelty really spans, again, a lot of understudied environments. This was spread around studies and samples that came from a range of populations, often in the very old or very young that have been less well studied, often in geographic areas that have been less well studied. Many of these, as we'll see on the next slide, came from international microbiomes and from really across the tree of life. Many, but as we'll see, not all of these were coming from less well-studied phyla or clades across the set of human-associated microbes. And so, there we go. And so this novelty really did come from several different areas. The biggest source is in understudied populations. So human populations either in the gut or across the body where microbes haven't been well sampled. Something we saw as far back as the HMP1 is that the sort of adult westernized human gut has been well studied enough that under many circumstances, you can explain 75, 80, sometimes 90% of sequences that you see from a typical in that sense, gut microbiome. And with the addition of new sequences from this study, we're able to push that up well over 90% in many cases. So we can explain now with references most of the reads that we see from a typical gut microbiome and often from other body sites as well. Except that when we go to non-westernized or international populations, the microbes that are carried tend to be sufficiently diverged that with previous references, we only get about 40 to 50% direct explanation, nucleotide level matching. And there's other things that we can do with protein functionality that I won't show here. But just with the addition of these newly assembled, surprisingly high quality genome bins, we can push that number even in international populations up to more like the original 75 or 80%. So with this single meta-analysis, we're now starting to much better cover human-associated microbial diversity even for less well-studied populations in this point, in this case. Oh, I think now my pointer's trying to die too. So a lot of this novelty comes from these understudied populations, but not all of it. Something that we've seen as somebody who looks at the human microbiome using reference genomes a lot, something we've seen over several years now is that one of the most prevalent organisms in probably everyone in this room, or 80 to 90% of the people in this room, is an uncharacterized subdilogranulum, so just one genus within the firmicutes. Surprisingly, when we looked at this clade in the assembled rather than reference sequences, it turned out that this subdilogranulum is actually probably 7 or so different species level organisms that separate very nicely phylogenetically. You get really clear divergence between all 7 clades. They're reasonably closely related to subdilogranulum, but not quite. And in fact, only one of them has a very recently sequenced isolate that's from the Gemiger genus, again, closely related to subdilogranulum and some of the other firmicutes. And these clades together are about 80 to 90% prevalent in all of the populations that we looked at. So these are very prevalent organisms that just haven't been isolated, haven't been sequenced. They're not rare, they're not particularly unusual. They just happen to escape that part of the tree of life so far. And we can see this type of novelty using the assembled data as well. And then finally, even in well-studied clades like the Bacteroides, which are arguably the most prevalent clade in the westernized gut, there are many, many isolate genomes available. But even for genus Bacteroides, we can find sometimes thousands of additional genes in the pan-genomes associated with new assemblies of organisms within these clades. So in this case, we're finding diversity that's must be functional somehow, but it's associated with bugs that have been well-studied in well-studied populations. So there's a range of different types of microbial sequence novelty that can be captured using, even in this case, a fairly simple whole community assembly-based approach. And we're working now on taking these results and sort of closing the loop to use newly assembled sequences as better references and integrate sort of assembly-based and reference-based analyses for the microbiome generally. As far as methods for studying and characterizing some of this novel sequence, we've just finished work on capturing recent lateral transfer events in human-associated microbial communities that help to sort of explain where some of this novelty comes from, what starts to drive bugs to diverge and personalize in their strain composition within each individual. And there have been lots of great studies of lateral transfer in the human microbiome, typically using isolates, where it's much, much easier to isolate out individual whole or near whole genomes and find recently laterally transferred elements in individual bins. But of course, some of the very earliest human microbiome work was in anecdotes of identifying lateral transfer in whole community culture independent data and then validating it, in this case, in isolates. And for anyone who doesn't recognize the article, it's one of the early metagenomic examples of finding a porphyrinase for seaweed degradation transferred from putatively an ocean bacteria to a gut-resident bacteria, specifically in a Japanese population. So we know this is going on, and we wanted to be able to sort of see it at scale in culture-independent data, and especially to capture recent lateral transfer events unique to specific bugs in specific people using whole community metagenomics. So one of the patterns the lab often follows in order to tackle a question like this is to put together a method that we can then generalize to do this in other settings, often with an excellent name that I never come up with. So in this case, the group put together a method called WAAFLE, which stands for that, in order to identify recent lateral transfer events. And by recent, we mean not captured in a sort of ancient event that appears in isolate genomes. It tends to be unique to a particular microbial community. So a very brief overview of how this works is to start from a single metagenome. Again, do a sort of lightweight single sample assembly, like we saw in the example of a few slides ago, that generates contigs. We don't even need to bin them in this case. So you'll end up with a very messy metagenomic assembly with lots of small to medium-sized contigs, and that's okay in this case. That's specifically what we've tuned this method to deal with. We do use a large external reference of pangenomes calculated for each microbial species. So we group together all the genes that have ever been known to associate with each reference isolate species and do a little bit of quality control to clean those up. And then you can either provide gene calls or identify genes within these metagenomic contigs and assign them to one or more homologous sequences in this pre-calculated pangenome. What this lets us do is really the main part of the WAAFLE process is differentiate between contigs that are sort of well explained by just one organism, and I'll say more about that in a second, versus contigs that just can't be explained by a single organism, even taking into account things like flaky metagenomic assembly. And then, of course, we can classify single organism contigs to their taxon of origin versus multi-organism contigs as potential lateral transfer events. And the key criterion that we use to differentiate between these two is basically to say, if we can explain a whole contig, even at a very, very lenient threshold, using just one organism's pangenome, then that contig probably came from that bug or something closely related that we just haven't seen yet in our reference. If we can't do this even very leniently, and we can very confidently assign multiple organisms to that contig, then it might be a lateral transfer event. And we start by doing this at the species level and bubble up to detect sort of further and more phylogenetically distant lateral transfer events. So the secret sauce here is, if you can explain a contig even very generously with one bug, we're done. Otherwise, if you find two bugs or more very confidently, then it might be a lateral transfer. So we've done a lot of computational validation for this that I won't talk about today to really tune it so that it has a very, very low false positive rate and a reasonable recall. Depending on the distance of the phylogenetic distance of the lateral transfer, we can get somewhere between a 40 and 90% recall with a fraction of a percent false positive rate. And applying this to just over 2,000 metagenomes, again from across the body in the HMP1 data, in the HMP1 population in this case, we get just over 110,000 potential new confident lateral transfer events. And we know we're undercounting these in order to prevent false positives. About a third of these are intragenous, so species level lateral transfer events that we're still very confident about. We can assign directionality to about 10% of them, which means we have a long enough contig to know who's the donor and who's the recipient. And this averages out to a handful of confident intergenous events per bug. So this means per bug, per microbial community, there are at least this many de novo lateral transfer events that are more or less unique to that environment. So this is supporting earlier work in isolates that say, hey, there's a lot of lateral transfer and new personalized lateral transfer going on in each human microbiome. And in the context of something like spaceflight, presumably affecting the nearby built environment as well. What this looks like in the gut, when we just summarize down kind of who's transferring to who, is that you can see a lot of phylogenetic structure. We've summarized this at the genus level here. You can see little clumps where, for example, Bacteroides and Parabacteroides will tend to transfer elements to each other. A Calibacterium and Subdiligranulum in this case, or likely the group of related bugs that I showed earlier, tend to transfer elements to each other. So there's a lot of phylogenetic structure, but that's not the only driver. You can see there's other sort of peaks and valleys in this set of who's transferring to who. And this is even clearer when we show this data as a network, either undirected, looking at all of these predicted lateral transfer events, or directed, using just a subset where we can get a confident direction. In the gut, the major distinction is by phylum. Firmicutes tend to transfer to firmicutes. Bacteroidetes tend to transfer to Bacteroidetes. And you can see even at a high level, there do tend to be, as expected, particularly hubby organisms. There are more plastic organisms that participate in more lateral transfer events. But really interestingly, if we quantify this by looking at the network, it is very scale-free. The rich do tend to get richer. There are a few bugs that tend to be the main drivers of which bugs are receiving lateral, or how many bugs are receiving lateral transfers. But which bugs receive the bulk of those are very specific. So even bugs with many lateral transfer partners, very plastic genomes, tend to have just a few lateral transfer partners that account for most of those events. And we can quantify that by differentiating between what seems to drive hubbiness or frequency, which is abundance. If you are a more abundant bug in an ecology, you tend to participate in more lateral transfer events. And we've limited this down to just the ones where we're very confident about directionality and participation. But the bugs that you tend to transfer with are driven by phylogenetic similarity. So even if you're a very plastic organism, you're abundant, you tend to participate in lateral transfer events with many different bugs. Just a few of your phylogenetic nearest neighbors tend to be the recipient of most of those lateral transfers. So this starts to tease apart how these two different, sometimes competing drivers of lateral transfer can shape LGT events in a community. And all of this is true not just in the gut, but across the body. Again, I won't get into some of the details today, but all of these properties and a lot of the phylogenetic and functional structure are shared between events that we predict in the gut and events that we predict in other body sites' metagenomes. We have looked a good bit at which functions tend to transfer, and we're starting to work on how and making some predictions about what different mechanisms are driving mobility in these cases. And we've gone in and validated a subset in as much depth as we can. We really want to tune out false positives in this context. And so in most of the cases that we've looked at, we actually have a hard criterion that forces there to be even metagenomic read coverage across the entire putative lateral transfer and confident support at the junction points. And we've taken about 20 or so of these and actually PCR'd across the putative junctions, and in almost all cases get a successful PCR too. So we're not just seeing these by sequencing. This is one example where all of that was true with a known mobile element integrating into a locus of unknown function actually. And this is pretty representative that elements of unknown function are statistically enriched both in transferred elements and the elements, the recipients of those transferred elements. And this is another case where we get very, very confident both read support and PCR support for transfer of an element of completely unknown function. And I've been showing the support for these over here. About 18 out of the 20 loci that we followed up with PCR primer design and validation from the original samples worked, many of which were again domains of unknown function. This is by itself not terribly important, but as a lab that's mostly computational, I love the chance to show a slide of all gels in this case. So it's mostly working, both computationally and experimentally. I think for the sake of time, I'll skip over some of the other vignettes from the HMP2 if the slides are available. These slides summarize sort of all the data for for a couple of individual patients, and make the point that either by zooming way in or by zooming way out and sort of summarizing all of the molecular interactions in the host and microbiome across the entire population, you get new sort of insights at either end of that scale. So I think the HMP2 by itself, and as we'll see in a few slides, in the context of spaceflight microbiome, makes an argument for really deep focused studies of the microbiome and host responses as a way to understand what's going on here. And then finally, I promised I'd at least touch at the end on some of the interactions of the microbiome with the built environment. And a few years ago now, we started to look at this in the context of the Boston subway, the MBTA, as an environment with lots of human environmental microbial transfer, just to see, hey, how are bugs getting from person to person rather in a community rather than in a microbial community? And are there whole cell sort of live microbial transfers of non-pathogens down in the microbiome of commensals that we wouldn't normally see? So we've done some work on that, and very quickly, as many in this room know well, very quickly ran into the issue that in the environment, you aren't really sure which bugs are alive and which ones are dead. Started to apply high throughput methods to assessing which microbes are viable in the built environment and which ones are getting there from humans in that environment. And very quickly ran into the issue that some of these methods are challenging to transfer to the microbiome and whole microbial communities. So examples like Propidium monoazide staining coupled with sequencing work very differently in our experience when you're looking at an entire environmental or human-associated microbial community versus individual organisms, and when you're trying to do whole community sequencing versus sort of targeted PCR, targeted PCR of individual organisms. So we sort of took a step back and are about midway through now a process of validating PMA as a whole community viability assessment for human-associated microbes in the built environment. And we've been doing this sort of systematically at the bench and then systematically computationally as well to compare there's a fairly small number of published whole community PMA-based viability assessments to say not just which human-associated microbes are detectable in a built environment, but which ones are still living there. So I'm happy to talk about this more offline. It's very much still ongoing now. To wrap up, I wanted to call out a few points that I think have been mentioned between today and yesterday, but maybe not called out as much yet and are interesting things to talk about later. I described the HMP2 today in part because I do think that a very sort of focused and probably long-term closed system study of the microbiome would be very interesting at this point. It would be extremely challenging to actually do in space, but using an Earth-based proxy would be completely doable using the kinds of resources and platforms that are available now. It tells you a lot about individual microbes up to whole communities, transferring between individual people up to whole human communities. And I've been surprised that we haven't gotten to talk much about immune parameters yet. I know that's on many people's minds, but especially in what we've heard about the spaceflight and astronaut microbiome, I think will be important to highlight more over the next day or two. Going back to the HMP1 especially, and speaking of the healthy microbiome, in such healthy individuals, it is hard outside of the context of a pathogen to really push the microbiome in a very bad direction. It tends to be ecologically resilient, but it might make an interesting target for sort of preventative or health maintenance measures. And I think somebody just said this earlier today as well. So I think, Iran, if that was you, I agree with you. And then we've also mentioned from the planetary protection angle, contamination a couple times. It was already mentioned to be essentially inevitable, but with things like what Nick just showed and Julie mentioned the other day, I think there's an interesting opportunity to focus on containment and tracking. That's some of the methods, I didn't talk about it today, but some of the methods that Nick described are starting to extend to whole communities as well, not with as much sensitivity, but with increasingly comparable specificity for whole community tracking of individual strains in addition to pathogen tracking. So hopefully those will come up during the later conversation breaks. And if my pointer survives one more slide, anyone whose face you saw on the slides is someone from the lab who did the work, and I especially want to call out collaborators. Nicola, I mentioned, Romnick works with us on a bunch, and especially on the HMP2. Rob and Morgan were key parts of WAAFLE, and Owen and Noop have been fantastic throughout both HMPs and manage the HMP Data Coordinating Center. And now I'm definitely over time, so thanks everyone.